Hi everyone, welcome to the Tropic Marin channel. We're here in Karlsruhe in the wonderful Natural History Museum. We also had a look at these magnificent aquarium facilities that are run by Hannes, the curator. So I'm really thankful that we can be here today and have a look around. But some of you may have noticed that behind us right now there's no aquaria, no corals, no water. Right now we want to look at the facilities up here. It's a lab and I want to chat about what's happening here. And with me is Dustin, who is in charge of the genetic studies that are being conducted here in the lab. But a little bit of background, I would say, information. Here in the museum, there's a lot of research going on, right? You have lots of different research areas.
Departments in the background. So we have mycology, we have entomology, we have botany, zoology in general. So there's lots of stuff going on.
Yeah, and long established research came out of this institution here. So it's a really high level scientific institution. And now corals are also finding their way into the genetic studies here. And this is what is happening here. So genetic studies means you extract the genetic information, the DNA of the organisms, in this case corals, and you try to isolate it and pick certain parts of millions and billions of genetic information. Then you have to try to make sense of that and how to pick the areas you want to look at with the background of identifying coral species, basically, right?
We have like a almost fully equipped lab, so we are a little bit limited, but we're still expanding. And we try to provide the service of genetics like to all departments and maybe also to external institutions to know a little bit better what kind of stuff they have and they're working on and to identify organisms.
So the process here basically is to take tissue of corals, like the corals you have here in the aquarium. Some of them have been here for decades and are doing really well, and it's a high diversity. So you take the tissue of the organism, then you kind of dissolve the cells so you can access the DNA that is there in the nucleus and in the mitochondria and the zooxanthellae and everywhere. You run it through different machines like the centrifuges, and you clean it to remove everything that is not DNA. Then you can take the DNA and decide which areas you want to look at. And these areas are then millions of copies, so a lot of material that you can then have sequenced. So you do sequencing as well?
Essentially, we're doing like the sequencing reactions. So as I said, we're a little limited. In principle, we can do it like all the way, all in-house here.
Yeah. The machinery that you can see here, you can use for basically all DNA work or genetic work. So you don't need a different machine for analyzing coral DNA. So you can use it for the entomologist, the mycologist, like everyone can use these machines, which is great because these machines are not very cheap. It's great if a lot of different departments can use the machines and generate data that can be used then for conservation, for describing species. The general process and the goal of why to do it is to know what organisms live in the aquaria, but also in nature. And that is a very hot topic at the moment, I would say. Like coral genetics is not something that has been long established and is being just done, right? It's still in development basically as we speak. So there's a lot of groups around the globe working on optimizing methods, on optimizing databases, all that. So you can actually identify coral species to a species level. And there's a lot of changes at the moment. Also like genus of Acropora is gonna probably expand into a lot of more species. So right now, I'm not sure, like 150, 180, something like that, but we'll probably look at beyond 500 because it turns out the more detailed you can look into the genetic information. I'm not a coral geneticist. I don't know how to say, okay, the species ends here and the next ends there, or what characteristics have to be met to say, okay, this is a different species. But it seems like a lot of acroporids are very regionally distributed. So across the Pacific, some islands have their own species, right? And then you get into the conservation aspect. If you have that just on that one atoll, then you're one hot summer away from extinction, possibly.
Yeah. It's especially interesting because like I'm also not an expert on corals, but to have corporations which have the expertise, which have the data, you need a good reference library. And it's especially interesting not only in corals, but in other stuff as well, that you look at genetics and the morphology and the ecology, like at the same time, in order to identify them to a species level. Yeah.
Yeah, always try to assess the organism as a whole. And then moving on, it will be very useful in the future to know what kind of species are in public aquaria, which aquarium has which species, and now that we can start to reproduce them sexually in artificial environments, could start to create a database or the knowledge of which species is where. So ultimately, like it's done with a lot of different other species that are endangered. So you can maximize the capacity of the ex situ coral culture in a more managed way, that we can make the best use of the facilities, like great facilities as they are here, to maximize their usefulness for conservation going into the future, because that's going to be an important topic. Yeah. I'm really happy to have been able to have a look around here. I'm sure it's been interesting for you. See you next time.
Bye-bye.
Take it easy. Bye-bye.